अस्सलाम वालेकुम स्टूडेंट्स, आई होप यू आर ऑल गुड. आप सब अच्छे से होंगे. Today we will discuss a very important topic, that is hemostasis. Before I will discuss hemostasis, cardiovascular system, there are two aspects. If we will just see here blood vessel, one aspect is blood vessel is normal. This is a blood vessel. This is endothelium. It's quite normal. There is no injury in the blood vessel or to the endothelium of the blood vessel. It's quite normal. The second aspect is when there is a damage to the cardiovascular system or to the blood vessel. Say for example, here it is an injury, here it is a puncture in the blood vessel. So here is a puncture, here it is a site of injury, here there is a damage done to the blood vessel or to the cardiovascular system. What we will see here, what we will observe here, as soon as the blood vessel is damaged or its wall is punctured or there is a site of injury here, there is a deposition of a semi-solid mass. Semi-solid mass is deposited here, and the components of this semi-solid mass, they are derived from the blood. All its constituents are derived from the blood. So this semi-solid mass which is deposited here, what it will do, it will plug this site, it will plug this puncture, it will plug this injury. And once this plugging takes place, the loss of blood, that is the blood loss, is limited. Blood loss stops, or we can say it is limited. So this mass, this semi-solid mass, is deposited at the site of injury, deposited at the site of the puncture, and once this is deposited there, this puncture gets plugged and there is no blood loss, or the blood loss gets limited. So to prevent the loss of the blood, this is what is called as hemostasis. That's what our topic is. Hemostasis means prevention of the loss of blood whenever there occurs a rupture or a puncture in the blood vessel. So definitely blood loss will be there. In order to stop or prevent the loss of the blood, and this prevention is achieved due to the formation of a mass, a semi-solid mass here, which will plug this puncture, and all its constituents are derived from the blood, and this limiting or stopping or preventing the loss of blood is called hemostasis. Fine. But equally important is to prevent the formation of this semi-solid mass, or to prevent the formation of this clot in the healthy blood vessel. When the blood vessel is healthy or it is absolutely normal, when this endothelium is absolutely normal, when this endothelium is absolutely healthy, there should be no deposition of this mass, no semi-solid mass or no clot formation should be there. There should be no clot formation. There should be no deposition of this clot or this semi-solid mass there. Now, it means that if there is no injury, if there is no trauma, if there is no puncture in the blood vessel, there are mechanisms going on. This is endothelium which is continuously resisting the formation of clot. This is endothelium which is continuously resisting the formation of this semi-solid mass, so that there will be the smooth flow of the blood, because there is no need for the clot formation, because the blood vessel is normal. So mechanisms are here operating, continuously mechanisms are here operating to prevent the formation of this semi-solid mass here, or to prevent the formation of the clot inside the blood vessel. So both these mechanisms are equally important. When it is healthy, mechanisms are on to prevent this mass deposition. When there is injury to the wall of the blood vessel, mass should be deposited, clot should be formed to achieve hemostasis, to prevent the loss of the blood. So there are two aspects. We will pick first this one, and later on we will see it another video lecture, how healthy endothelium prevents, resists the clot formation or the deposition of a semisolid mass. So here we will discuss hemostasis. But before that, all those substances which work here, which prevent the clot formation, we call them as anticoagulants. We have a term, we call them as anticoagulants. Got it? And here we will find here those substances which work to form this semisolid mass or to form the clot, we call them as procoagulants. So procoagulants work during the vessel injury, during the damage or trauma to the cardiovascular system, and anticoagulants, they are working continuously in our cardiovascular system to prevent the clot formation. So mechanisms are always there to resist the clot formation, right? So there are two aspects. So let us first see this one, hemostasis. So I repeat, hemostasis means basically to prevent the loss of blood. And when there is a loss of blood, when there occurs the oozing of blood from the blood vessel, of course, when there is an injury, when there is a puncture in the wall of the blood vessel. And to prevent the loss of the blood, and this prevention we call it hemostasis. And now this hemostasis, it is achieved in many steps. Mainly there are three important steps to achieve the hemostasis, to prevent the loss of the blood, of course. Number one is vasoconstriction. Let me write the steps. Hemostasis is achieved. Number one is vasoconstriction. Vasoconstriction. Number one is vasoconstriction. And number second step is platelet plug formation. Platelet plug formation. This must be plugged. This puncture must be plugged. The injury must be sealed. And the third is, that's very famous term, that is coagulation. Coagulation is entirely different from the platelet plug. There's a difference. We will get to know each of these steps involved in the hemostasis. So vasoconstriction, number one, platelet plug formation, and there is coagulation. Let us see the first one, vasoconstriction. Why should vasoconstriction occur at the site of injury? Why should it take place at the site of the puncture? We must know it. We must know why it should be there. Vasoconstriction definitely will decrease the blood flow towards the site of injury. This we have to achieve. Means that if there's a blood vessel puncture, if there's a blood vessel injury, then what should happen? There must be vasoconstriction, so that the blood flow towards the site of injury, so that the blood flow towards the site of puncture, it must be decreased to achieve hemostasis, to prevent the blood loss. And now this vasoconstriction, how it is achieved? क्या इसका mechanism है? कैसे हम vasoconstriction को achieve कर सकते हैं? Let me discuss it here very clearly. This is here vasoconstriction. Vasoconstriction is here. How we achieve it? Listen. At the site of injury, injured nerve endings release vasoconstrictors. That's very important. Release vasoconstrictors, and definitely it will lead to what? Vasoconstriction. Second, damaged or injured smooth muscle. The wall of the blood vessel, it too contains smooth muscle. Smooth muscle contracts. Damaged smooth muscle, injured smooth muscle contracts, and this contraction will also lead to what? Vasoconstriction. Two ways. So we can say this is neurogenic vasoconstriction, and this is myogenic vasoconstriction. And there is one more way to achieve the vasoconstriction, to limit the loss of blood, to prevent the loss of blood. And what is that? The injured endothelium, very important point, injured endothelium, it releases a chemical substance called endothelin. And this endothelin is a vasoconstrictor. Yes, of course, it is a vasoconstrictor, and it will directly lead to what? Vasoconstriction. So there are at least 3 ways to achieve the first step, that is vasoconstriction. Number 1 is injured nerve endings. What they will do? They release vasoconstrictors and they will lead to the vasoconstriction. What do you call it? You call it neurogenic vasoconstriction. Second way, smooth muscle contracts, damaged or injured smooth muscle also contracts. Where is the smooth muscle present? It's present in the wall of the blood vessel. As we already know, tunica media, it is containing a smooth muscle layer there. So smooth muscle contracts, and this way it will lead to the constriction. This is called myogenic vasoconstriction. And the third way to achieve the vasoconstriction is injured endothelium. Injured endothelium, what it will do? Injured endothelium will release endothelin, and endothelin itself is a potent vasoconstrictor, and it will lead to what? Vasoconstriction. And overall effect, overall consequence of this is what will happen due to this vasoconstriction? It will lead to decreased blood flow towards what? Towards the site of injury. So the blood flow will decrease towards the site of the injury. This is basically what is to be achieved through the first step, that is vasoconstriction. I hope it's clear. Number second important step of hemostasis is, number second step we will discuss here. It is called as platelet plug formation. As I have written earlier, platelet plug formation. Platelet plug formation. Now the platelet plug formation, how it is formed? Injured endothelium, the injured endothelium, it secretes a very important factor that's called as von Willebrand factor. Where from it is released? It is secreted by what? Injured endothelium. It releases a very important factor, von Willebrand factor. And this von Willebrand factor, let us first see here. This is here blood vessel which has got damaged. ये मान लीजिए blood vessel है जो damage हो चुकी है, ठीक? This is its endothelium, right? And say for example here the blood vessel has got damaged. So here the blood vessel has got, this is the puncture point. And now this injured endothelium, what it will secretes? It will release what? Von Willebrand factors. And this von Willebrand factor, it is acting as an adhesive for what? Platelets. It is acting as adhesive for platelets. Means that von Willebrand factor has a receptor present on the platelet surface. So definitely this is a blood here. Platelets will come and they will sit on this factor, because platelets have a receptor. Platelets have a receptor for what? For the von Willebrand factor. Say for example this is a von Willebrand factor V shaper, and these are the platelet cells. They got just stick to what? Von Willebrand factor. So platelets basically they rest on the subendothelial layer. This is an endothelium and this is subendothelial layer. So platelets now rest on the subendothelial layer due to the von Willebrand factor. That's the reason इसलिए हम इसको बोलते हैं it acts as an adhesive or a sort of a sticky substance for what? Platelets. So here is a layer of the platelets formed. This is called as, this aggregation of platelets, it is called as platelet adhesion. We call this as platelet adhesion. How it is achieved? Due to the von Willebrand factor which is released by injured endothelial cells, and this von Willebrand factor, it has a receptor on platelets. So the receptors are present on the platelets So platelets are going to get stick to von Willebrand factor, and this coming together of platelets, we call it platelet adhesion. तो इसको हम platelet adhesion बोलते हैं बेटा, ठीक है. Now platelet adhesion जब हो गया, इसके बाद there occurs platelet activation. First let the platelets come together and then let them get activated. Now this platelet adhesion, it will lead to the platelet activation. What I actually mean that, listen, I will show you here. This is for example a platelet cell here, right? And say for example this is a receptor on the platelet surface. So there's a platelet cell here, right? Platelet cell, and this is a receptor, and this receptor, it is meant for what? Von Willebrand factor. So for example there's a von Willebrand factor here, von Willebrand factor. This interaction between the receptor and the von Willebrand factor, it will lead to what? Platelet activation. Now what does platelet activation here means? It means at least two things. यहाँ पे दो चीजें होंगी. Number one, there is a release of thromboxane A2. Thromboxane A2 is released by the platelets, and number second result of this activation is degranulation. Degranulation means that platelets do contain granules in the cytoplasm. They just secrete their substances out of the cell. That's what's called as degranulation. Mainly talking about alpha granules and the delta granules. There is also lambda granule which has a lysosomal activity, alpha granules and delta granules. And second important aspect of activation is that is the release of thromboxane A2. Thromboxane A2, it has two jobs to do. Number one, it is itself a vasoconstrictor, and vasoconstriction is required here. I did mention in the first step. So first it will act as a vasoconstrictor, and second very important function that it is playing, it is acting as platelet aggregator. It is a platelet aggregator. What it is going to do, ये क्या करेगा, ये platelets को attract करेगा. It will just aggregate the platelets already present or on the already present platelet layer, because already the platelets have come on the subendothelial layer due to the von Willebrand factor. Already we have achieved what? Platelet adhesion. Now platelet adhesion will lead to the platelet activation, and platelet activation will release what? Thromboxane A2. What it will do? It will aggregate. It will further attract the platelets, so that now the platelets which are coming, they will now get sticky to the platelets already there. Are we clear? There are two aspects. First layer of platelets, they are not on the platelets present on the non-platelet surface. That is platelet adhesion. Now the second batch of platelets which are coming, or the third batch of platelets which are coming, they will rest on the platelets. So the platelets now become sticky to each other due to the presence of what? Thromboxane A2. So it's acting as a vasoconstrictor. Number second role is platelet aggregator. Now another aspect of the activation is a degranulation of what? Alpha granules, and there is delta granules. Now we first look what is the composition of alpha granules and what is the composition of the delta granules here. Let me show you here alpha granules and there are delta granules. Delta granules, what they contain? Delta granules, what is the composition of the delta granules? What is present inside? Number one, there is calcium. Number second, serotonin. And number third is ADP. A, D and P. Alpha granules, what they contain? They contain two important factors here. Number one, platelet derived growth factor, and number second is a protein you already know, fibrinogen. So alpha granules contain PDGF, platelet derived growth factor, and number second component is there, that is fibrinogen. So delta granule composition, calcium, there is, yes please, serotonin, and there is ADP. Now we will first see serotonin. We already know serotonin क्या करता है बेटा, ये भी vasoconstrictor है. So vasoconstriction is again here achieved. So vasoconstriction is a very fast reply to what? To the injury of the blood vessel. So serotonin again, it is what? Vasoconstrictor. Now what's ADP doing? Yeah, ADP, it binds with the platelets. It is released by the platelet, by the delta granules, and once ADP is released, what ADP is doing, ADP is actually platelet, it is a platelet activator. Yes, once ADP binds with the platelet, it has a receptor on the platelet surface, and once it binds with the platelet, expresses more and more receptors. Platelet expresses more and more receptors. For what purpose? For von Willebrand factors. So for example, here a receptor which is present on the platelets, and once ADP binds with the platelet becomes more and more expressive, it becomes more and more activated, and more these steps will occur, and more platelet adhesion, right, and more platelet activation, and activation will lead to the thromboxane A2, which will lead to the platelet, more and more platelet aggregation. So more and more platelets are going to come and get sticky to each other. So ADP is a platelet, of course, activator. Serotonin is a vasoconstrictor. ADP activates platelets. Let platelet to express more and more what? Receptors. For what? Von Willebrand factor. So this step is repeated again and again, right? With new platelets, which are just relaxing, which are just relaxed. They don't know that injury or some trauma has taken place. But once ADP comes in contact with the platelet gets activated. So these steps are again repeated. So more and more addition, more and more aggregation. Got it? Now serotonin is a vasoconstrictor. Now calcium, we already know calcium is a very important factor in the coagulation mechanism, very important factor, and it is here released by the delta granules. कैल्शियम का रोल क्या है बेटा? कैल्शियम का इंपॉर्टेंट रोल यहाँ पे ये है, कैल्शियम होल्ड्स, दैट्स वेरी इंपॉर्टेंट, इट होल्ड्स coagulation factors together. It holds the coagulation factors together. Remember that, because we know that coagulation in coagulation, one factor leads to the activation of another. So they must be brought together so that one activates the another coagulation factor. For that purpose, the coagulation factors, they are pinned by what? They are held by the calcium. So calcium has a very important role. It holds coagulation factors together. Later on we will see in the third step, that is a coagulation. वहाँ पर भी हम देखेंगे कैल्शियम का बहुत इंपॉर्टेंट रोल है. हम बहुत सारी जगहों पे देखेंगे मीडिएटर काम करता है ये. लेकिन what exactly it is doing, it is holding the coagulation factors together. That's a very important thing. So for example, if factor 10 is there, factor 11 is there are many factors which are acting there. So holding them together so that one will activate the another factor. So it holds the coagulation factors. ADP, platelet activator. Serotonin is vasoconstrictor. Now this is PDGF, platelet derived growth factor. We will see the role of the PDGF in detail later on, but here we'll tell you platelet derived growth factor. Number one, its role is to stimulate the proliferation, to stimulate the division in the smooth muscle, because we know that in the blood vessel, जहाँ पे blood vessel का rupture हो गया है, ये मान लीजिए blood vessel has got ruptured here, I did mention earlier, it is intact. So for example, this is the site of the injury here, and now here's a smooth muscle. Now smooth muscle proliferation has to take place so that the damaged smooth muscle cells will get replaced by the new ones, and to stimulate the smooth muscle division, platelet derived growth factor plays a very important role. Another important role is it also attracts fibroblasts. Fibroblasts are the connective tissue cells. It attracts fibroblasts towards the site of the injury. And fibroblasts are the cells which basically are the factories for the formation of collagen. Fibroblasts will come at the site of the injury and will synthesize more and more collagen, because we know that these cells, these endothelium cells, they rest on the basement membrane, and the basement membrane is actually made of, yes please, it is made of collagen type 4. So let me repeat it here again. We'll tell you again here what basically is happening here. देखिए बेटा, see, it's a blood vessel here. Say for example, it's a blood vessel here, right? And here say these are the cells here. This is one cell, say for example it is another cell, and this is another cell. So this is the site of the injury, Now the fibroblast PDGF, platelet derived growth factor, will attract fibroblasts towards the site of the injury, and once they will come towards the site of the injury, they will lead to the formation of this collagen. Collagen is very important because we know that epithelium cells, they rest on the basement membrane, and this basement membrane is made of collagen. So for that purpose, fibroblasts, they have to come so that the collagen gets deposited here. Right? So two important rules I did mention here about the platelet derived growth factor. Number one is to attract the fibroblasts towards the site of the injury so that fibroblasts will synthesize collagen, and collagen here gets deposited again. Number second is that it stimulates the division or proliferation of what? Smooth muscle, so that the damaged smooth muscle will get replaced by the new ones. So this is the role of platelet derived growth factor. Fine. So now get back to the topic. So we are discussing platelet plug formation. I will repeat. Injured endothelium, what it is releasing? Yes, please. Von Willebrand factor. Von Willebrand factor is adhesive or it is sticky. It will bind with the platelets. Platelets do have the receptors for the von Willebrand factor, and this platelet aggregation is called platelet adhesion. Now this platelet adhesion will lead to the platelet activation, and platelet activation, that is this interaction between the receptor and the von Willebrand factor, which lead to the platelet activation. Now this platelet activation, yes, it has two aspects. Number one, I did mention, that is the release of, yes, thromboxane. I'm repeating the things, thromboxane A2. And number second important rule is the, yes, degranulation, alpha, and there are delta granules. Alpha granules, yes please, fibrinogen, I mentioned about the role, fibrinogen, and number second is PDGF, platelet derived growth factor. And what is the composition of the delta granules? Yes, three important things I did mention. तीन चीजें मैंने बताई बेटा इसके बारे में. Number one, serotonin release is there, and number second is calcium, and number third is, yes please, what is this number third? ADP. Got it? So PDGF mentioned the role, proliferation of the smooth muscles, and attracting the fibroblasts towards the site of the injury for the sense of the collagen. और serotonin पहले ही बता चुका हूँ, vasoconstrictor है, calcium holds the coagulation factors together. And what is ADP? ADP is platelet activator, so that these steps will be repeated again. And thromboxane A2, two roles. Number one, vasoconstrictor, and number second, it is a platelet aggregator itself. Right? Now, all these steps, platelet activation up to this, what will happen after that? See, I will show you here what will happen here. Okay, I will rub this. See what's going to happen here. Now this is a blood vessel again. Let me draw it again here. Fine. And now here it is, these are the cells. Say for example, these are the cells here. यहाँ से भी हम बनाएंगे एक दो cells, और यहाँ से बनाएंगे. So these are the endothelial cells, and now we already know here was a von Willebrand factor. Got it? And sticky to what? Platelets. So this was platelet adhesion, and now after this, more and more platelets are coming here. You got it? So this is a sort of is a big aggregation of the platelets. A big aggregation of the platelets is here. So this big aggregation of the platelets, this is what is platelet plug, and this is actually called a primary platelet plug. So this will result into formation of primary platelet plug, but this plug is loose. It's loose. Here we complete platelet plug formation through this mechanism. So two important things here: platelet adhesion, and then there is platelet aggregation. Platelet aggregation will result into formation of primary platelet plug formation, and which is of course loose. And here we complete this platelet plug formation. Now after that, there is a third step which is taking place, and that is called as coagulation, and that we will discuss in the next video. You should revise these two steps first, and tomorrow we will be discussing coagulation and further two processes associated with the coagulation, two pathways associated with the coagulation, that is extrinsic pathway and there is intrinsic pathway, right? So you should revise these things so that you will understand the next topic, that is coagulation and the two pathways of the coagulation, that is extrinsic pathway and there is intrinsic pathway. Till then, Allah Hafiz, see you in the next video.